the FDA approves something on 20 patients, the rest of the world thinks that that's not very good, but the drug manufacturer won't go build more evidence because they got their US approval. If we could sort of have a
Yeah.
clear understanding what needs more research and truly what doesn't. This is gold, let's just switch. And I think the data are all there in RWE. We need to do a much better job of getting it into the hands of the regulators in a usable fashion.
Hey everyone, and welcome to What's with RWE. I'm Kostas.
And I'm Lester. Thanks a lot for joining us for this is technically the opening episode for the new season, our second season of What's with RWE. You can see that we're actually recording this just before the holiday season. That's why we have our essentially our decor. And I do apologize, in the previous episode, we were unable to record the videos. That was really my fault. So we're trying to at least rectify that a little bit here. So today we actually have a very special guest with us today. We have Dr. Natasha Leighl. Many of you probably already know of her, but she's on our podcast really to talk a lot about RWE, but a little bit about Natasha. She is currently a medical oncologist, or has been a medical oncologist for a long time at Princess Margaret Cancer Center. She's also the OSI Pharmaceuticals Foundation Chair of the Princess Margaret Cancer Foundation. She's also a professor at the University of Toronto, and she probably has many more titles as well. But we can't go through that entire list because we're trying to restrict this to only an hour episode from now on. That's at least what our producers have told us. But Natasha, welcome to the show. Thank you very much for joining us today.
Thank you both so much. It's a treat to be here.
Excellent. Just before we jump right into the conversation, I know this is right before the holiday season. So any plans for you for the holiday season? Anything of great interest that you're gonna partake in, or the usual family dinners?
Nothing of great interest. I think the usual chance to see family and friends and perhaps unwind for a day or two.
Excellent. That's great. So we've had actually Dr. Jerry Battis on. So Jerry, who's also our chief medical officer, on, and we asked him this question. I want to ask you the same question: is how did you become an oncologist? And the reason why we asked that, well, at least my curiosity around that is, when I was at ASCO this year, every single person I talked to who's in the medical oncology field or a medical oncologist, incredibly intelligent, incredibly capable. So there's a huge surface area of things that you could have done. So why become a medical oncologist? It seems like a field that you deal a lot with death, you deal a lot with some of the darker sides of not necessarily humanity, but of just reality. So why did you become a medical oncologist?
Well, thanks for asking. So I actually started young, and I would say it was serendipity. When I was 16 in high school, we had a chance to do a research project, and our school was attached to the University of Toronto, and there was a poster on the wall in the university section that was advertising a research project in biophysics. And that was when I first met Professor Dick Hill. He was a radiobiologist and senior scientist at the Ontario Cancer Institute, which was connected to the Princess Margaret. And I worked with him for the first few summers. Terrible at bench research, but not only did I get to meet him and some amazing physicians and scientists who became oncologists, but also greats like Dr. Ian Tannock and Dr. Fifi Liu all had labs and did research at that time. So that was my initial start. Then when I was in medicine and doing my training, I really fell in love with the human aspect of oncology. That was a time when really chemotherapy and radiotherapy were what we had for patients. And despite that, patients were grateful. They were grateful that we cared, that we tried, and that their lives and their quality of life matter to us. So it was very rewarding, much more rewarding than seeing other patients in the emergency department. And so later I went into oncology, and I've been so grateful because really oncology has made extraordinary progress, really bringing bench and bedside closer together. There's been so much scientific advance, even in the 20 plus years that I've been in oncology, that when I started in lung cancer, chemotherapy was very exciting, and now chemotherapy is what we want to avoid. We're really interested in precision medicine, so tremendous things, and really a watershed time, I think, in oncology.
Great. No, that's it seems to be that's a common theme. At least Jerry as well. I think he also mentioned it was a research project where he was mentored by someone who essentially inspired him to take part in this world. So it seems to be a little bit similar for yourself as well, Natasha. Great.
Yes, yes, it was. And I've had many mentors along the way. Dr. Francis Shepard is my lead mentor in lung cancer. My start in RWE actually, though, didn't come from Dr. Shepard. Does a lot of clinical trials, but is also very interested in translational research. It actually came from Professor Martin Tattersall, whom I worked with. He was very interested in communication with patients, what patients understood. And that really introduced me to the whole world of health services research and outcomes research, which I found very fascinating. I think drug development is critical. I think clinical trials are critical in that translational aspect, so we can understand the biology of the disease and how to make an impact. These are all essential. But the other things that were really important to me was, how does the system work? Are our patients treated fairly by the system? Are there things that we need to fix? Who are we as oncologists? How does our understanding and our behavior impact on our patients? And what needs to be changed to make things fairer and better, to make sure that everybody can take advantage of progress?
So so keeping that in mind, because I think that's a relatively unusual sort of a research focus for a lot of oncologists. I think when we talk to people, they're really laser focused on the therapies, on clinical trials, especially really trying to put through rigorous testing potential therapeutic options for patients. Health services is a little bit surprising. Is there anything about your approach to oncology that people might find surprising?
Thanks. I don't think so. At the time, when I started just over 20 years ago, health services research was not where it was at. I found it fascinating, the study of
Mm,
us, the study of the system, the study of patients outside of trials, that chance to really make things better for every patient. That really meant a lot to me, at a personal level, at the level of an oncologist and a healthcare provider. I will say in the last 20 years, now that's most commonly what people come to me with. People are interested in outcomes. When I
yeah.
first started, Dr. Shepard launched my career in pharmacoeconomics, which was very unpopular for most of my career. And now it's the big thing. So I kind of feel like I started all this when it was very unpopular and very difficult. But it's nice to see that it's so much greater interest. And there are many more people, making their careers and really making advance in this area.
That's terrific. Yeah, and I can empathize with you on that. I started on that same path probably around the same time, and it was I'd say not a hard slog, but it was really uphill trying to convince people that there was a complete other side to the health system that really needed examining if we were going to make it not only the most effective we could make it, but sustainable as well, which I think was the other piece that a lot of people started to become concerned about and really wanted to understand. So that said, there's a lot of change. You mentioned over the 20 years that you've been that there's been so much change in the way what we've seen, the progress in treating patients. If we were looking forward, we're at the end of the year already, and there's a lot coming next year, in the next year or two. What really excites you about what's coming down the pipeline? And that could be therapies, it could be in programming, it could be in testing. If we open it up, what's the most exciting for you?
Thanks so much. So I would say that crossing that bridge between the science and the trials and implementing things in the clinic to really optimize how patients do and feel is what is most exciting to me. I've been doing a lot more work with liquid biopsy testing, and this is very interesting. We have the ability to diagnose patients' cancer types, to direct
Hmm.
therapy, to diagnose resistance, and also figure out, maybe how much or how little treatment a person needs. If you have curative treatment, are you really cured or not? Do you need more? Did we give you
Hmm.
too much? Can we scale back? And the same in the advanced disease setting. How much is enough? Do you need more drugs? Do you need fewer drugs? Have we got the wrong drugs? And so I'm very excited about that over the next 1 to 2 years. We're doing a lot of work developing trials. But again, they're very practical trials. They're really aimed at everyday patients and improving everyday patient outcomes. What we've spent the last few years doing is really improving molecular testing on the ground. So, not just theoretical, but really showing what the value is in the clinic of precision medicine, and what it means to avoid chemotherapy and how valuable it is, both with tissue biopsies, liquid biopsies, genomics, targeted treatments, immunotherapy. So we've been doing a lot of work like that, and it's really been great to see it come to fruition, be able to offer that to patients now every day. And for me, my next short-term frontier is introducing liquid biopsy to the clinic. And then after that, we're learning that cancer, in medical school, we learned that cancer was clonal. And what I mean by that is that, there was one cell, then there were daughter cells, and they were all exactly the same genetically, epigenetically, phenotypically, they were identical.
Hmm.
But we've learned that is so far from the truth, and that neighboring cells can be completely different. And things that we thought, for example, somebody with an EGFR mutation, which is an important kind of lung cancer, couldn't possibly have anything else going on. And yet we've learned there are different kinds of EGFR mutations within the same tumor. You can see different kinds of mutations without EGFR in different cells, even in the same biopsy sample. And so I think for the next 10 years, that precision, that ability to look at single cells within the tumor, that'll be the next thing that we have to say, okay, the science and the trials are great. How do we bring this to patients? How do we make it practical? How do we make it real world? How do we make sure that at the level of every patient, not just the select few in trials, we can really do this thorough diagnosis of cancer, resistance, treatment planning, and also optimize the timing, right? Maybe you don't need everything at the same time. Maybe you need some things on day one, some different things on day two or day three. So that's those are my short and long term things that I'm really excited about.
Well, I have to take the opportunity to ask you this because you are the expert. Actually, for those of you who aren't familiar with Natasha's work, Natasha is basically the expert at liquid biopsy, right? I mean, globally, I would say even globally well known for this. She was the lead for a trial called Nile, which really kind of pushed a liquid biopsy testing into the forefront. So I have to take the opportunity to ask you this, Natasha, is maybe you can help define liquid biopsy for our audience because we're just assuming there's billions of people watching and maybe not everyone understands what that is. So maybe if you can just give a little bit primer, at least to you, what is liquid biopsy? Because it is quite nuanced. There's quite a few applications of it. So if you can maybe do us a favor and give us a little bit of a definition, that'd be really great.
Sure, happy to. And I have to say that I've had so much fun, and we've been able to help so many people with liquid biopsy. It's really driven my enthusiasm and a lot of the work that my team and amazing other teams around the world have done in this space. So, a liquid biopsy, we most commonly use it to refer to a blood test where we look in the blood and we can find DNA or genetic material or RNA or other things, fragments of DNA that are from broken down cancer cells. So we can use that potentially instead of going in and taking a sample of the tissue with a biopsy or cancer tissue. We can use that to make different types of diagnosis, genetic diagnoses, to look at different things that are associated with the cancer cells that are now in blood. So, in the old days, my patients used to say to me, well, isn't there a blood test you can do to see if the cancer's back or not? We're still not quite there, but we're pretty close. And sometimes I can tell my patients whether or not their cancer's back with blood test. So, there are also other kinds of liquid biopsies. So Dr. Jeff Liu, who's one of my colleagues at the Princess Margaret, is doing some really exciting work looking at what we call breathomics. So you breathe out, he collects all the little droplets, and he tries to do testing on that to see, are you at risk for lung cancer? Could you have a lung cancer? or not? And then if so, can we find out anything else about that? And so there are many different approaches like that. We heard about a team in BC a couple of years ago who could pick up like lung cancer alterations in people's urine. So really any liquid will do. Blood is the easiest. And, certainly in my research team, people would much rather collect blood than all the other things. So we can use it, to some extent for diagnostic purposes, to some extent in this precision medicine. Does your cancer have mutation X? And so I can match you to treatment Y. So that's really launched. And so studies like the Nile study and our value study that we did here in Canada have really shown the benefit. We find more alterations for more patients, more people get better targeted therapy and can avoid chemo, live longer, and do better. And some of the costs, particularly for drug treatments, are actually less by getting the right person to the right treatment at the right time. You are quite right, though, Lester. There are other uses. So in addition to that sort of diagnostic or precision medicine use, we can find out when treatment's no longer working, and we can also find out, for example, if somebody's had surgery, if they're at high risk of the cancer coming back, we may find genetic material in the bloodstream. And so that's something called minimal residual disease. And we're currently doing trials trying to find that in people with the earliest stages of lung cancer to figure out, right now we just do surgery and people are followed under surveillance, but maybe these people need more treatment. And so we're actually doing a big study in Toronto across 3 hospital sites with some amazing surgeons. And they are helping us look at patients, look at, whether there's any of this cancer DNA in the blood after surgery, and then can we escalate and give them treatment to get them to cure where surgery has not. So lots of potential uses. Also, even in screening, I told you a bit about breathomics, but maybe there are blood tests that can say this lump in your lung is cancer or it's not, or it's a very likely cancer. So we should do a biopsy in you versus, it's probably not cancer. It's just living in Toronto or other exposures you had in your lifetime. We shouldn't do a biopsy for you. We should watch. So lots going on, lots of potential.
Great. And actually, liquid biopsy was not one that I would have. That was not surprising that you mentioned that because you're the guru in that space. One area that I'm surprised that you didn't mention because you recently wrote a paper around NLP, around the use of NLP for real world data mobilization, and how that was very quite accurate compared to just manual curation, which is a lot of what's done actually today. I'm curious what your thoughts around. So you didn't bring up that component like NLP or of course the big word artificial intelligence, right? Those, like, you didn't bring that up as something that you see down the line for the next 5 to 10 years. How do you think about that,
Yeah.
at least that corpus of tools?
It's a great point, Lester. And, I probably should have mentioned something about the work we've been doing in AI. I kind of feel like it's already here. It's already everywhere. We just have to learn how to use it. And I think some of the technology will improve, but I think it's here. And so because I'm so interested in the outcomes, I kind of don't think of how I get there. But, we had this great opportunity to work with this team. They're from a company called Pentavere, and there are, of course, other teams that do great work with AI. And what they've developed is normally when we would do outcomes research or we would try and collect real world data, get an army of summer students and fellows and trainees, and people go in and they collect the data from the charts and they write everything down, and we have to cross check everything, and everyone has to type it the right way in the Excel sheet, and the statistician feeds through everything, and it's a lot of work. And so in this work that we did with Pentavere, they were actually able to plug into our medical record system, extract all the data, and populate the database all on its own. And it took, it did take us a week or two to develop the algorithms and develop the coding and the right language. They had to fine tune the database a little bit to really optimize capture of the data the way that we wanted it. But it didn't take, an army of 10,000 summer students in 5 years to put together, a 20,000 patient database. It was actually really easy. Just kind of flip the switch and it all just comes out. So we actually did a study. I thought that was great, but I was a little suspicious. And I thought, well, how do you know the computer's as good as people? So we actually did that. We got this army of summer students that collected all the data. One of our fellows, she's now in Sherbrooke in Quebec, Marie-Pierre Gauthier, she supervised and she compared what the students put together versus what we gathered through the natural language processing. And it was highly concordant. Complex things that might have only gotten mentioned in the notes once or twice natural language processing, or AI, was much better at finding. So medications, things that the doctors forgot to list, they were very good at that. Other things that were a little more challenging were, these sort of descriptive terms in medicine. So the lymph node was slightly enlarged versus the lymph node clearly had cancer. These were sort of where the computer got a little confused, and so sometimes it would say, okay, it's cancer, and oh gosh, we don't know. Whereas the students might be able to cross-reference that somewhere else, or be able to make some other interpretations. So, by and large, for I would say 98% of the data we collected, the computer was or AI was as good or better than human or manual curation. There are a couple of things where, we still have to fine tune a little bit. But the real fault is actually in the medical records we keep, right? Because we don't do them systematically. It was always unstructured data where, a bit more of the art of medicine and sort of no discrete or firm terms for things. Where things were clearly structured or, had clearly firm data, AI outperformed people pretty consistently. So I mean, I thought that was great. We're trying to move to using teams like Pentavere now to gather a lot of our data. The other great thing about Pentavere is, if I do a study last summer and we want to publish in 2 summers, I have to go back and update everybody's information. And so that requires another army. The students have to go in. We have to double check everything. With the natural language processing, you just hit the button and boom, everybody's records are caught up. So it really is fantastic. And that 1 or 2 week investment to make sure that you're collecting what you want and you fine tune your algorithms is so worth it. And it's so much faster and I think more accurate than a lot of the manual curation we do. So I think it's amazing for real world evidence. I think we should all just do it. I think we should make sure that we structure our health records so that you can get perfect data capture and use machine learning to really generate big data sets. Jeff Liu, whom I mentioned before, one of my colleagues, he's trying to harmonize data sets between different hospitals and cancer centers across Canada. And they're also trying to use natural language processing and artificial intelligence to kind of harmonize things. And then, if I publish a data series of 1,000 patients, well, that's great. But when you have 200,000 patients and you can keep their outcomes updated, that's incredibly powerful. Then you can start to see things in the data that you'll never find in a clinical trial, and you'll never find in these sort of small single center RWE reports. So I think this is the real potential. And I think for us to really maximize the use of RWE, we're going to need natural language processing or NLP and AI or machine learning. A very long-winded answer to your question.
No, that's great.
Yeah, no, absolutely. It offers a world of opportunity, but going back to this sort of the old world versus the new world of clinical trials versus using real world data. When you have this, how do you overcome that level of skepticism that may still exist? I mean, I'm sure you deal with it when you are publishing or trying to make changes to treatment strategies with data that doesn't necessarily come from a clinical trial. What's the strategy to overcoming that skepticism?
So, it's very interesting. When I started, there was a lot of skepticism that these data would be worthwhile, but we've been very lucky. Researchers have asked very good questions. So I'm mindful of a nice publication from Chris Booth in Kingston, who does a lot of health research outcomes work. And so he looked at, how long after surgery could you delay starting preventive treatment or adjuvant chemotherapy and still get a good outcome? So that was actually really practical. And that was an everyday issue that was just not addressed by the current clinical trial literature. So I think this is where we have to go, right? When there's a practical issue that's not addressed by current evidence, that's where real world data and researchers need to go. So for example, recently we've learned that if you give immunotherapy to people with cancer and then you switch to targeted therapy, maybe you didn't get the results in time, maybe there weren't opportunities before, whatever reason, patients are at higher risk of immune side effects for the first 3 months of treatment. We didn't see that in the trials. We actually saw that from a real world publication. So they presented their data. This was a very nice French study looking at patients with KRAS mutant lung cancer. They showed that in the first 3 months, patients had higher rates of immune-mediated toxicity. Then they went back and they looked for it in the trial and they found it. And now it's a thing. It's a big safety signal. We use it routinely. And it was discovered through real world evidence, and now everybody's finding it in all of their series with all of the drugs. But we never looked at it before. And even, on data safety monitoring committees that I sit on, we all ask the question, well, did they have immune therapy, and what's going on with these patients versus other patients? Because now we know from real world evidence that there are differences that were not reported in trials. So that's what I would say to skeptics, that there's so many questions that are unanswered, and look at the things we've been able to do with real world evidence. I think that there's much less skepticism today. I think people have embraced it. People are interested. For example, one of the societies that I work very closely with, they have a great journal, the Journal of Thoracic Oncology. They used to publish a lot of outcomes data, then they kind of got a bit too expensive. Now they publish mostly interventional trials, but they recognized that these are important studies. So they actually created a whole new journal, JTO Clinical Research Reports, and it's really for real world data, for us to be able to pool and share some of these unique insights that you're just not going to get from clinical trials, and that are so relevant to patients and healthcare providers in everyday practice.
So the other topic you sort of touched on was the topic of scale, or the need for the numbers of patients to really be able to identify these very distinct insights, and insights that, especially when we start linking them to biomarkers, you start to slice the salami smaller and smaller to do these analytics. How do we get sort of all the centers across Canada to start actually pooling data, to actually commit to sharing? Because I think there've been a number of attempts of doing that, and so far we haven't seen the success that I think we'd all like to see. What's been your success to working with others across Canada?
Thanks. I think it's because we are colleagues and friends, and we have so much in common. One of us will have a trial, we'll send patients to that center, or vice versa, and so we already collaborate in other forums or fora. So, right now, Dr. Jeff Liu, again, he's leading this our group called Karma Bros. I didn't pick the name, but anyway. And what they're trying to do is they're trying to build those links between provinces. So there aren't these silos between provinces. And so the hope is that through that grassroots effort, we'll be able to combine and have a pan-Canadian database. And then the provinces will say, hey, these guys can do it. We have even better data. Why can't we just do it? So we're hoping that at least that level of cooperation will inspire others. And of course, there's a lot of work that's ongoing. Your team at Exactis, and I think many others, are trying to work across different institutions across the country to try and build those links, and to try and have resources where people can pool their data. I mean, they do it in the US, things like Flatiron or CancerLink. There are a lot of different ways to pool data. I think one of the important things is that as we pool data, in addition to volume, quality is so important. So we need to be sure, again, that we have as many structured, accurate fields as possible that are merged. You need to make sure that you don't say it with small caps in one center and all caps in another, and therefore it's thought to be something different, right? All these little nitty gritty things that you just don't think about, from an analysis perspective, all of those things I think need to be aligned. And so I think, some of the work we're doing at the ground level is to try that amongst our databases, things we think are interesting. And then our hope is that, whether it's drug manufacturers who are collecting data, Health Canada and pharmacovigilance, and institutions and provinces who collect a lot of cancer data, that they will also start to align, try and harmonize their data fields and pool for more strength, and more ability to detect, important nuances in the data.
So just for the audience, maybe this would be helpful, because you brought up the topic of quality, harmonizing, and structured, right? Are those the same thing? And what does that mean, and why does that matter? Actually, just for the audience, so that just in case there are individuals out there that are not familiar of how real world evidence studies are conducted, perhaps this would be helpful for that.
Sure. So, I think that they're all similar. They ensure a good product. I think for people who are familiar with systematic reviews and meta-analyses, people often say, garbage in, garbage out. If you don't have any studies that are good to pool in the first place, you don't get a very good pooled result at the end if all the studies were flawed or the data weren't collected properly. So the same is true with real world evidence. You want to know that when you're asking a question, the answer that you get is as close to the truth as possible. So, for example, let's say I have a database and a colleague has a database, and I've collected smoking data and I've done the work to make sure that it's as accurate as possible, checked with patient report, had double verification, whatever it is, and somebody else has not done it. Then when we pool that and we talk about the outcomes for smokers, only half the data is actually useful. So then you have to say, okay, what are we going to do with the missing data in the other database? How are we going to do the analyses, and what are the strengths for combining versus splitting? Can this other team go back and add smoking data? Did we use the same definitions? I call a non-smoker someone who's had less than 100 cigarettes in their lifetime. Maybe they call a non-smoker something different. And so then if we're looking for biologic trends, we want to make sure we identified and define things the same way, so that we can then merge our data sets and they can be reliable. So that's sort of what I mean by sort of quality. You make sure it's accurate, and as detailed as possible. Harmonizing, make sure that you're measuring the same thing, you're defining the same thing, you're expressing it the same way. So that ability to harmonize. That's sort of what I mean by quality and harmonization. So they're very similar, but they're all about making the data as similar as possible in terms of how we collect, how we define, how we report, so that we can pool it and come up with a good answer. None of this garbage in, garbage out.
Great. No, absolutely. Thank you very much for that. So now with this high quality data that theoretically that we have, let's just assume that in the future, this beautiful world where we actually collect this data first and then subsequently be able to harmonize it in a very structured database, so that we can then do, as you suggested, analyses on it. And it's very clear from your example with the immune therapy and the certain mutations that then subsequently there is maybe not the best approach, right? And that was found in real world data. So that has a clear clinical impact. Now, you talked about real world evidence in terms of a health services impact. And that's something, a major reason why you got into conducting real world evidence studies. So maybe could you speak a little bit more about that component, like making a health services level impact with real world evidence? And the reason why I ask this is primarily, this may be a poor example, so you can completely reject this and and give a completely different example. Is right now, let's say certain types of therapies, when they come into Canada, they need to go through various approval processes, right? And most of them basically require clinical trials. It's like phase 3 clinical trials to be then subsequently widely accessible to patients. And real world evidence is not necessarily that it's not looked at, but is not the primary driver for, let's say, access to therapy. So can you speak about how real world evidence can shape health services, and maybe where there's still additional opportunity for it to help shape health services as well?
Sure, Lester. And I think that's a great potential use of RWE that is really underutilized by regulators. So I'm actually going to give you 3 examples that sort of highlight kind of different aspects of this whole issue. So the first example I'm going to give you is understanding, or the first 2 is understanding how a treatment performs in the real world. So uh, when I first started my career, we had a targeted therapy, a drug called erlotinib that targeted, later we learned it targeted patients with EGFR mutant lung cancer. It targeted their cancer. But at the beginning, we thought it was good for everybody because we didn't have very good treatments for lung cancer. So it was a little bit better if you used this, and it didn't matter what kind of lung cancer you had. So that was approved. It was a big Canadian advance. It was used very widely. And then over time, when we discovered the EGFR mutation, most people gravitated towards using these targeted therapies for that. But then there was always this belief that, probably still does a little something in people who don't have the mutation. So we actually did a study. Dr. Kirsten Pertuzé, she's now an oncologist at William Osler. She's phenomenal. So she did the study and she used data, real world data, from ICES or ICES, which is the provincial database on health outcomes of patients here in Ontario. We also collaborated with Cancer Care Ontario. We got a list of everybody who'd had erlotinib in the province over a 5 to 10 year period. And we looked at patients who clearly didn't have the mutation. They're on the mutation, they're on the treatment for a very short period of time versus patients who clearly did. And in these databases, mutation status isn't so good. We need to work on that. But so we sort of looked at sort of, if you're only on it for a short period of time, is it still improving your outcome like the clinical trial said it would? And we found the reverse. We found that these people actually, if they never got erlotinib, they actually lived longer than these patients who clearly didn't have a mutation that got erlotinib. So that was interesting to us. And it kind of clearly reinforced this is something we shouldn't be giving to someone without mutations. Now, could you argue it's not a prospective randomized trial in this group of patients? Yes, you could. But in the US, this has lost its label. I think in Canada, if it hasn't lost its label yet, it's certainly gone out of use. But we think that we've provided some pretty good data for whom not to treat with these drugs. In Germany, they were using this. They'd abandoned it briefly, but I think they're going back to it, where if you approved a new drug, you could use the drug for a year. The company could market the drug. The German government would pay for the drug. Patients would use it. And at the end of the year, they would look at patient outcomes and they would compare it to the current standard or comparator. So let's say second-line treatment, patients would give them docetaxel chemotherapy, which is kind of awful. And then you'd have this new treatment. And then they would look at, okay, how did people who got docetaxel do sort of in the recent past versus people who got this new drug? And based on how that drug performed, so let's say it was safer, people didn't go to hospital, people lived longer, well, that's good. So you could charge more. If it was worse, you had to charge less. And if it was about the same, you didn't, and you couldn't prove any clear advantage, then, potentially you'd have to price it the same. So that was a very interesting application of real world evidence. Now, the problem was they couldn't get the data in time. It was a real challenge, but they've really upped their game in terms of RWE, national database collection and centralization. And so, they're much more automated in real time. So I think they're actually going back to that system. So that is potentially very helpful in Canada, right? So, if there's a blockbuster drug, but we're not sure that it's really helpful, could use it for a year and see what happens to these patients. The third part of that story is, for example, in lung cancer, there are these small populations with targeted therapies. Nobody can do a randomized trial, or the companies won't. It takes too long. And we recently, for example, did a study in patients with RET fusion positive lung cancer. So we had great data, 80 or 90% response rates. People were on treatment for more than a year. Amazing drugs. They would go to the brain and shrink brain tumors. Fantastic, right? In my opinion, clearly better than chemotherapy. The regulators said, oh, you don't know how well chemotherapy does in this kind of lung cancer. You've shown us the overall data, but we don't know about this group of patients. So, in databases like Flatiron, and in this Canadian database, national database that's being put together, you can pull out all the RET positive patients and say, okay, this is how they did on treatment A, treatment B, treatment C. It's not perfect, but at least it's a type of benchmark, right? So you can say, okay, if you had the mutation versus if you didn't have, what's the difference? Do these patients really do better, or they all kind of do about the same and badly? And then the regulators can say, oh, okay, so just based on this, indirect comparison, this is how the new treatment does. So it does enable. Now we are not, as people who should be generating these data, we're not publishing it fast enough. We're not analyzing it fast enough. We're not producing it in a consumable fashion for the regulators, and we need to start doing that. So that's one thing. And then the regulators need to start using it. So instead, we had to do this huge randomized trial where the targeted therapy for RET just blew chemotherapy out of the water. And it was honestly ethically inappropriate to have let this study continue, right? Because we already had the data. Now, in fairness, chemo did work a little bit better than it does in people without the RET fusion, but nowhere near as well as the targeted therapy worked. And so then how do we go back and teach the regulators, okay, these are the thresholds. You don't need a randomized trial for this, this, and this, but maybe you need a randomized trial for these other things. And then we also teach the drug manufacturers and everybody else and the trialists so that we steer our resources correctly and we don't end up where, the FDA approves something on 20 patients, the rest of the world thinks that that's not very good, but the drug manufacturer won't go build more evidence because they got their US approval. If we could sort of have a clear understanding what needs more research and truly what doesn't. This is gold, let's just switch. And I think the data are all there in RWE. We need to do a much better job of getting it into the hands of the regulators in a usable fashion.
No, that's great. No, thanks for those examples. I was actually, I wasn't aware of the German example. That's a very kind of a very interesting way to use or apply real world evidence. I believe I actually, you can correct me if I'm wrong. I'm not familiar with this as much, but I believe Italy does something where a lot of the, I think, oncology based therapies they have to go through some sort of process similar to what the German example is in Italy, I believe, for a lot of cancer therapies. But I'm not actually sure
Yeah.
about that.
Thanks. Yeah, I haven't heard a lot lately. I think it would also be challenging in Italy to put those data together. They do have a lot of expanded access programs, so I do hope that the regulators are using the data from that. I think Italian investigators work hard to get those data to patients and get those drugs to patients and collect the data. I haven't heard. There's a lot more cost sharing, and there are many more innovative schemes for getting drugs to patients. I think in Italy, I think they're a real leader in that.
Yeah, and I think getting the data in hand in time to be able to make quality decisions. I think that's the key for everyone, right? It's the sort of the golden sort of moment in time to be able to say, how do we ensure that we're making good decisions with good data instead of just pushing out that decision and saying, well, we don't have the right data, or we don't have Canadian data, or whatever market you're located in just trying to ensure that we have infrastructure in place to be able to access the data that we have in hand. I think that's one of the that's what everyone is trying to do right now. So
I
I
agree.
don't know where Canada ranks at the
I mean,
moment.
I think we're emerging, but it starts at home, right? I need to make
Yeah.
sure that the data on my patient has the relevant endpoints. Did the cancer
Hmm.
shrink? Do they feel better? Am I measuring it in an objective way? Did the cancer grow? When did it grow? You would think that these things are easy to find in the medical record. They are not. And so we need to make sure that we collect those data in a structured way, as objectively as possible, and automate it, right? I mean, we have this tremendous power of AI and NLP. Um, Automate it. Get all the data in an easy to collect format, hit the button and go. I think that's what AI has given us. I think we're just not using it.
So, Natasha, does that mean when you are entering that data, you might think about it a little bit differently today than you might have 5, 10 years ago, because you're thinking, am I putting it in a way that I am going to be able to utilize it with these tools in a year or two or three?
Absolutely. And I think the problem is that after I have done the chart review or the data collection, I forget. And then I just go back to my old way of doing things. And so it's really to have those structured elements that are there every time, but to also make them easy. Sometimes it's, you put in 50 symptoms and you have to click, 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 and none of them have to do with your patient. There must be some easier way for data light collection for the essential things that are easy for us to do as part of our normal practice without making the patient wait an extra 8 hours while we fill out our database forms. So
Yeah.
some easy ways to have these structured things in the system, auto populate. I'm kind of hands off. I just put in the stuff that the patient, I, and others need to know, but all those other data that are so essential to assessing how the patient is doing from a regulatory standpoint are there. And I don't see why we can't do it, right? I mean, increasingly we have these new healthcare systems that, in theory, talk to others. So I think we should be doing more. Eh, Having said that, human nature is very powerful. Like I said, like I remember after I've done it, and I'm really good. The other day I was making sure that everybody had the exact dose and the dates that they were on each dose of these drugs, because otherwise it's very hard to find these things.
Yep.
And by next week I'll have forgotten, and I'll be doing my usual. So important to make it part of the routine.
Yeah, no, I completely understand where you're coming from. It's like me and recycling. In Vancouver, like, it's supposed to be out of the goodness of your heart, and you want to improve the environment. And I'm not trying to get political here, but then sometimes you just forget. I'm just lazy sometimes, and I throw that banana peel and where I shouldn't throw it, right? But then, of course, the Vancouver city government said, okay, if you're going to do that, then you get a fine. So then when you get the stick, you're like, oh, you know what? I should recycle this. So I completely understand where you're coming from. So you talked about a couple of things, and we're going to wrap this up because we don't want to take up too much more of Natasha's time. Natasha's obviously incredibly busy. She saving patients and trying to change the world. So this is so appreciate you being on the podcast. But you mentioned a couple of items I wrote down. One of which is, in order for real world evidence really to make a bigger impact, it needs to be easy to collect, or some easy way to make it high quality. You said the answers need to be fast enough, and then you said it has to be in consumable fashion, and then it needs to be used by the regulators. So if you had, since it is the holiday season, and we're going to wrap things up here, is, if we gave you one wish, and Kostas is the genie, and he was able to just, snap his fingers, and then you get one wish. Like, what would you wish? What would you put in place that, and you only get one, right? What would you put in place that you think would really improve cancer care for all cancer patients?
Oh gosh. Well, Lester, that is a real challenge, and that's about 50 wishes. But I'm just going to take a tiny RWE slice about that. I'm not going to talk about prevention or early detection or cure of uncurable. I'm just going to talk about my RWE stocking. So in 2024, what I want CADIF, I want the regulators, PCPA, all those dudes to come together with investigators and people who generate databases and hold databases. We get together, we agree what's needed, how we make it consumable, and put it in the format that can be readily generated and readily used for policy decisions, and then we do it. But I think we have to have that meeting. So if Santa's only going to give me one thing to advance outcome, I want that meeting. I want us to figure out how to make it work from top to bottom, and then build it in 2024. So that's that stocking. I hope I get more stockings and more wishes.
Well,
I'll see
absolutely.
I'll see what I can do with my snap. There we go. Done. Expect the invite in your inbox. Well,
Great.
thank you again, Natasha, for spending the time with us today. We do truly appreciate it. We appreciate your insights into all of the RWE and health services research you've been doing, and we continue to look forward to the evolution in using actually liquid biopsies in place of what we have today, or adding to it, and giving us new dimensions of the abilities to care for patients. So thank you for your time. Lester, I'll give the last word to you to close it out for us.
Great. Again, of course, thank you very much to the audience for watching the first episode, of course, the first inaugural season of What's with RWE, and now tuning in for the second season of What's with RWE, and to our fantastic guest, Natasha, for joining us today. Really starting things on a very, very high bar. So now we have to make sure that every single other episode can follow up with this. So that's sometimes the double-edged sword there. So thank you very much. If you did enjoy this episode, please comment below, subscribe, hit that notification bell, and have a great day. So thanks a lot. Appreciate it.